Welcome to Goal 3. This one is all about the cell. The first module here is about the membrane and how things move across it. Hopefully after listening and learning this information, you'll be able to explain the basics of cells, describe in detail the cell membrane, probably draw it on a test, describe the functions of proteins in cell membranes, and the methods of transport across that cell membrane. What is a cell? It's not just a round ball with a nucleus in the middle. They can be any shape and size. We have trillions of cells, but bacteria are made of just one. The cell is considered the smallest unit of life, so unless you're at least one cell in size, you aren't life. Cells do all the work, and drugs work at the cellular level, so I think cells are the coolest part of AP, even though most people aren't really keen on them. I hope by the end of Goal 3 you'll at least appreciate their spectacular abilities. Here's a good average cell that the authors stuck every possible thing an animal cell might have in it or on it, and they put it here. There's actually no cells that look exactly like this. The cell membrane is probably the most important part of a cell. Without its working and doing all its jobs, the cell would just die. The membrane is the only barrier it has between it and the whole outside world. The smallest unit of life is the cell, and although we're made of trillions, losing even 1,000,000 doesn't even kill us. But many organisms, if they're only one cell in size, that membrane is really important. If it's punctured, ruptured, whatever, it becomes non-functional, the cell can die. This picture shows a basic cell membrane, and they're eerily alike between all organisms. Plants, animals, bacteria, protozoa. Your cells have very similar cell membranes to everything else. Practice drawing this out and label the parts, the phospholipids, the integral proteins, the ones that go all the way through, or proteins on just one side of a lipid bilayer. Note that there are two layers of the phospholipids here, and embedded in that are small molecules of cholesterol. You can see the darker orange right here. That's the cholesterol. The next few slides will go over some of these components. For example, this is a representation of a phospholipid. The yellow structure is the hydrophilic or water-loving phosphorus part, and the blue tails are fatty acids. They are the nonpolar part, or the water-fearing hydrophobic part. The two long chains are fatty acids again. The one that's bent is an unsaturated fat, and the one that's straight is saturated. Remember, fatty acids are nonpolar, and therefore the two nonpolar parts will stick together. Here's another picture of a phospholipid. Okay, two fatty acids, or the phosphate is the red molecule there. If you mix the phospholipids in water, you shake them up, and then let them go, they'll actually form this lipid bilayer structure with the nonpolar blue parts together, and the water-loving part, the phospho part of the phospholipids, will be outside. Okay? Molecules with no charge are not attracted to water, and as a result, water molecules tend to push them out of the way, which is why they form this nonpolar region together. Okay? Hydrophilic is the charged part, and hydrophobic is the non-charged part. Okay. Here we see a cross-section of a cell membrane, and you should notice two different structures. The phospholipids are the round purplish structures here, and the fatty acids are the little darker green things. And the proteins are the lumpy structures within the membrane. The membranes are fluid. They bend, they move, which allows us to bend and move and not break our cells. If you'd like a quick link, pause the slideshow here and watch the short YouTube. It's a really good example and an explanation of the cell membrane. So the main components of a cell membrane are again the phospholipids, which is a lipid bilayer. There's two layers of phospholipids. Cholesterol, which helps keeps our membranes fluid, although too much actually makes them rigid, which is kind of weird. Sometimes there's attached carbohydrates and many proteins. The proteins have the function, just like we talked about in Module 2, and we're going to talk about some of those functions in the next few slides. One of the things the proteins can do is transport things across. These are carrier proteins or transport proteins. They bind and drag molecules through a lipid bilayer and release them on the opposite side. In some cases, the channel proteins are simply tunnels, okay, like this one. This requires no energy, but things can diffuse through it. If there is more of the substance on one side than the other, diffusion goes from high to low concentration until it reaches about equilibrium. Some things can diffuse right through the membrane. They don't need a transporter protein, but they have to be nonpolar because they have to get through that blue part here, which is the nonpolar region. So in other words, fats can diffuse across, but if this little square molecule is water soluble, it needs a transporter. Okay? The more specialized form of diffusion when it uses a transporter is called facilitated diffusion. Okay, and most carrier proteins are used for that. Another thing proteins can be in membranes are receptor proteins. These are proteins that allow the cell to receive instructions. A little bit like doorbells, some molecule, like a hormone depicted in green here, will bind the receptor protein on the outside of the cell and cause an action on the inside. If you ring a doorbell, you let the people inside know you are there, but you haven't entered the house. For example, insulin binds its receptor on the surface of the cell, and the cell reacts by letting glucose in. But insulin itself never does get inside the cell to make that happen. This ability to cause an action by binding to a receptor protein is called signal transduction. This will be revisited even in more detail when you get to the endocrine system and talk about hormones, as well as during the nervous system and neurotransmitter action. A third type of protein in cell membranes are marker proteins. Marker proteins extend across the cell membrane and serve to identify the cell. The immune system might use these proteins to tell friendly cells from foreign invaders. They are as unique as fingerprints and play an important role in organ transplant, for example. If marker proteins on a transplanted organ are different from those of the original organ, the body will reject it as a foreign invader. These proteins are also used in intercellular communication. Our immune system recognizes our cells as self and know not to attack it, while recognizing foreign cells and cancer cells as the enemy, and they do attack. Another thing that proteins can be are linkers or anchors or attachment points. Adhesion molecules are proteins located on the cell surface and are involved in binding with other cells or with the extracellular matrix in the process of cell adhesions or cell interactions. Essentially, adhesion molecules help cells stick to each other and to their surroundings. Adhesion molecules include integrins, the Ig superfamily cell adhesion molecules, cadherins, lectins, and others. Don't worry about you learning all these names. Just remember that proteins can stick cells together or stick them to the other substances around them. They play a critical role in a variety of biological processes. For example, one important part of adhesion molecules serve in the immune system to enhance pairing between receptors that are less active and their ligands and transmit that signal to an effector to induce the inflammatory response. So even tiny things will trigger an immune response. In development, they play a key role in tissue morphogenesis, cell migration, axon guidance, so how our nerves form. Pathologically, they have been found to play a role in tumor metastasis and invasiveness. So understanding these is very much of great importance for oncologists. Sometimes proteins can also be enzymes. Remember, enzymes convert substrates to products, and sometimes to hold them in place, they are anchored in the cell membrane. So in summary, proteins in cell membranes can act as transporters, linkers, receptors, or enzymes. The next thing we're going to talk about in this module is how to get things across a membrane. which we mentioned a few slides ago just briefly, obviously things need to get inside of cells and some things need to get out. Hopefully you can guess that nutrients need to move in and the wastes need to move out. So let's talk about the different ways things can go across the membrane. The first and simplest is diffusion. Diffusion is passive movement, which means no energy is required, from a high to a low concentration. All small nonpolar uncharged molecules like oxygen or carbon dioxide or anything lipid soluble can diffuse through lipid membranes. So simple diffusion, things just move directly across. But if you're not tiny and you're not nonpolar, sometimes you need a transporter. And when you use a transporter, even though you're doing diffusion from high to low concentration, it's referred to as facilitated diffusion. Facilitated diffusion still uses the concentration gradient, but a transporter is used. It usually occurs with molecules like sugars or amino acids, and sometimes ions. It can't diffuse across the lipid membrane and therefore can't get through the hydrophobic part without a transporter. Osmosis is the diffusion of water from high to low concentration. Water can go across the membrane, but very slowly, because remember, the middle part of the membrane is nonpolar and it doesn't like water. It's very important that you need to remember in this particular example that the only thing moving in osmosis is water. Sometimes it's difficult to figure out which side of the membrane the water concentration is the highest. So often the phrase "salt sucks" helps me remember this. Whichever side has more salt or sugar or any dissolved substances, that's the way the water wants to go. So if you put a cell in a bucket of really salty solution, the water from inside the cell is going to go where the salt is, which is out and into the bucket. If you put it in a solution of pure water, because there's more dissolved substances inside the cell, the water from the bucket will move into the cell. So here's a red blood cell in this top picture, and it is in a solution where it has equal salt and water concentration on both sides of the membrane. But in B, the solution is incredibly salty, so the water is moving out. The solution, because it has more salt, is referred to as hypertonic, and when the water moves out, the cell shrivels up. This will kill the cell. In the bottom picture, the solution is hypotonic, which means it has lots of water but no salt. The solution inside the cell, the cytoplasm, is saltier. The water moves in, it causes that cell to swell, and eventually it could burst. Both cases, whether the solution is hypertonic, more salty, or hypotonic, less salty, the cell is going to die. You want to make sure that the solution your cells are in is isotonic, equal concentrations on both sides of the membrane. This is why you can't give somebody pure water or super salty water in an IV. Please take a moment to pause this slideshow and click on the YouTube osmosis. It's only about 90 seconds long, and it's a great explanation of osmosis. There's also another one by the Amoeba sisters if you really want a great tutorial. Here's a quick animation of a red blood cell in a hypertonic solution, hypotonic solution, or isotonic. So the red blood cell goes in the hypertonic solution, the water goes where the salt is, it shrivels up. In a hypotonic solution, the cell has more dissolved substances inside, so the water goes in, causes the cell to swell, and eventually burst. It's best if you put blood cells in isotonic solution, equal concentration, so the cell stays happy. Another way things can move in and out of cells is filtration. So think of a filter. Why does filtering happen in a regular filter? So you put water above a filter, and it goes through the filter and catches anything big enough that the filter will catch. Usually it's done by the force of gravity or by push. In our bodies, we have some filtration pressure in our capillaries due to blood pressure. Because the heart is beating, it's going to create some pressure to move that blood along, and sometimes it's high enough pressure to push things out. So the humans, our force behind filtration is blood pressure. Hopefully your blood pressure is not so high that you push out too much, and that can be a problem. The last way that we're going to talk about things moving, or small things moving in and out of cells, is active transport. Active transport moves things at any concentration gradient, so it could move things from low to high concentration. The reason it can do this is because it requires energy. Because it's work to stuff one more person in an already crowded room from low to high concentration, you'd have to have something to push. The form of energy used is ATP, and it also requires a transporter. So in this example, in order to transport this molecule, the transporter itself finds the molecule, and energy changes its shape so it can push that molecule out, for example, in that picture. Proteins or transporters in the cell membrane do this. They're usually named for what they transport, and they require, again, energy. In summary, movement of small particles is done by diffusion, facilitated diffusion, or active transport. Osmosis is a specialized form of diffusion of just water. The ability of cells to control what goes in and out allows them to take in what they need and get rid of what they don't want. If you don't get rid of the waste, they could build up and be toxic to the cell. On the other hand, if a cell can't get in an energy molecule, it's not going to be able to generate any energy. This makes the membrane semi-permeable or selectively permeable, and without the ability to control what goes in and out, the cell would die. Selective permeability is the cell membrane's greatest asset. Sometimes very large particles must move across a membrane, and of course you couldn't make a hole big enough for a large particle without making the cell die. So moving large things requires the membrane blebbing, that is actually a word, in or out. And so moving something in is referred to as endocytosis, and generally membrane is lost, and moving something out is exocytosis, and generally membrane is gained. There's a little picture here. If you want to import these large purple molecules, there's a little invagination, they get trapped in there, you get a little vesicle, and it's taken to where it's used in the cell. If you want to excrete something very large, the Golgi will package it in a little vesicle made up of phospholipids. It will go to the cell membrane, merge, and release that particle out. Pinocytosis is a specialized way of taking in liquid, and phagocytosis is taking in a solid or food. Sometimes this is referred to as transcytosis because both happen generally at the same rate. The HIV virus enters cells by transcytosis, and they're not really in the cell, but they get across because they're inside membranes and then get into the blood and can invade other cells. In summary, the lipid bilayer is made out of phospholipids. It can have many different proteins, carbohydrate attachments, and cholesterol in it, but the main thing is the phospholipids. It is incredibly flexible, and this matters so that when we move or twist or our stomach growls or our heart contracts, we don't break our cells. Its greatest asset is its ability to be selectively permeable. What this means is it is able to control what goes in and what goes out. And cell membranes contain proteins, and the proteins have functions. Remember that some of these functions include receiving information, signal transduction, acting as linkers, cell adhesion molecules, for example, acting as receptors, connectors, and transporters. Don't forget, you have Google, you have your lab packet, and you have the web. And always ask questions if you don't understand.